Okay, so we continue. We'll take the question at the end. So I think we'll continue with Nathan Cross from University of Sydney, and he's going to talk to us about EEG, fMRI, and cognitive sleep deprivation. Thank you.
All right, I have a lot to talk about, so I'll dive straight in, but I just wanted to thank Tan and Christoph for inviting me to speak at this symposium today. I have no disclosures. And I'll start with a metaphor, because I love metaphors. If you can picture a healthy brain like a plant, and if the nurturing and development of this healthy brain has its roots in a large number of lifestyle factors, such as the food and drink that we consume, as well as the various activities that we perform. But importantly, also sleep has an important role in that. And sleep itself has many paths to nurturing a healthy brain. One of those is that sleep prepares the optimal conditions for cognitive performance. And there are some leading theories about how sleep maintains a healthy neural environment. You probably have heard of the glymphatic model, in that during sleep, the brain flushes out toxic waste through the perivascular space. Now, this is still controversial, but it's a theory. And another model of how sleep maintains a nice balance in the brain is through synaptic renormalization, or what's known as the Shy hypothesis, in that throughout the day, plasticity sort of saturates within the brain, and sleep is important for downscaling all of our synaptic connections so that we can relearn again the next day. But what I'm going to focus on today in this talk is something a bit close to home. So I'm recently a dad, and as you probably are well aware, I've not been getting much sleep. And so I'm going to talk about what happens when we don't sleep in terms of our cognitive function. And it's well known that sleep loss shows a dose-dependent effect on decreases in cognitive performance. But what happens in the brain? In terms of a neurophysiological and particularly an information processing framework, we're interested in understanding why, when we don't sleep, does our cognitive performance suffer day to day. There have been few studies that have looked at functional MRI in a sleep deprivation context. And this is one from about 10 years ago by Thomas Yeo with Mike Chi, where they looked at before and after sleep deprivation, and they showed that in the sleep-deprived state, the default mode network becomes more coupled with the attention networks. And importantly, participants at baseline who had a segregation of the default, a greater segregation of the default mode and the attention network, showed a resilience to the cognitive effects of sleep deprivation. However, this was only after global signal regression was performed in the pre-processing. When they didn't do global signal regression, they did observe that there's a widespread increase in connectivity in the sleep-deprived state. Now, in that study, they didn't follow that path further and investigate, but we were interested in looking into that. And so while I was a postdoc in Concordia, we conducted this experiment, this study, with a bidirectional model of sleep deprivation, where healthy controls came into the lab twice, once after after they stayed in the lab and had a full night of sleep, and another time when they were staying awake for 24 hours. What was nice about this experiment is they also had an opportunity to sleep, and then we could reassess their brain activity and cognition after a recovery period, so we could look at the bidirectional effect of sleep. This was all done with MRI and high-density EEG. And we probed different cognitive tasks in domains of attention, working memory, and vigilance. And across the board, we see that sleep deprivation leads to a deficit in cognitive performance. Now, first up, we were trying to probe the functional organization. So we looked at the primary gradient, functional gradients, to see if there was any change in sleep deprivation. And what we actually found was there's no real difference in the sleep-deprived state in the functional gradients in terms of their range or the distribution of brain regions along these gradients. At baseline, the functional gradients somewhat partially predicted resilience to sleep deprivation, but it wasn't very strong. So there's a cognitive performance decline in sleep deprivation. But there's no major change in the functional organization or the gradients of connectivity. But we did see that there's quite a widespread increase in brain activity or connectivity in the sleep-deprived state. So what's going on there? So in a follow-up study, we kind of used a hierarchical model of the brain from the whole cortex level down more and more clustering into different regions. And by using this hierarchical model and a framework developed by Melanie Bolly and Habib Benali of integration between and within systems in a hierarchical manner. So in this model, total integration of a system is the sum of the within systems or within network integration plus the between network integration. And you can do this at each level of the hierarchy. So for example, at the network level, the total integration is the sum of the integration of all nodes within a network plus all of the networks integrating with each other. And you can go down the hierarchy and do this. And what we found was in the sleep-deprived state, across the board, there was an increase in total integration at every level. The effect was a bit stronger as you went down into the more nitty-gritty clusters, but it was statistically significant across all levels. Now, the total integration did increase with sleep deprivation. However, it didn't really recover after the recovery nap. But if you remember that the total integration is the sum of this within and between terms, you can also look at the ratio of the two. Now, this is called the functional clustering ratio. And this ratio normalizes sort of the segregation to the level of the total integration in the system. And it can highlight a breakdown of functional connection between different networks. And interestingly, we found that the functional clustering ratio increased in the sleep-deprived condition, but also during a recovery nap, this went up a lot and then came back down in the post-recovery, suggesting there's a bidirectional effect here of sleep on this metric. And what this suggests is that in terms of brain segregation integration, there is a continuum from a well-rested awake state to the unconscious sleep state, and that sleep deprivation pushes the brain somewhere in between those two. Now, nicely, we found a strong relationship with cognitive deficits in terms of their accuracy and reaction time. And importantly, a recovery that was also related to the amount of decrease of this ratio to the improvement in cognitive performances. So we've discovered this continuum of functional clustering or segregation of brain activity, and we found that the magnitude of changes following sleep deprivation were related to cognitive deficits. But what might be the reason behind this? Well, we did observe, and these are just two example regions for visualization purposes, but we did observe an increase in the amplitude of brain signals in the sleep-deprived state, and that the amplitude was greater. And we look at this across the cortex, you see that it's not uniform, and in fact, this map correlates nicely with the changes in integration that we see. We also saw an increase in the global signal amplitude fluctuation in the sleep-deprived state. And as you see here, it's related to total integration in each three of the vigilance states, the well-rested, the sleep-deprived, and the post-recovery nap. But it's the strongest correlation in the sleep-deprived state, suggesting that the global signal is also involved in this change in integration. And why might these amplitude of low-frequency fluctuations be important here? Well, remember I mentioned those two models of how the brain is maintaining a healthy neuronal environment. So both glymphatic detox and synaptic renormalization have previously been linked to large amplitude slow waves during sleep. So when you're sleep deprived, there may be this increased homeostatic drive, which maybe starts some of these processes occurring even while you're awake, which may result in large fluctuations in the brain signals, which then starts to impair the functional segregation and integration ratios, which would lead to cognitive performance deficits. Now, this is a model that obviously needs further testing, but it's a nice model to bounce off. And if we want to look at maybe some alternatives to how this might work, we can leverage dynamical functional connectivity. There was a paper on dynamical functional connectivity in sleep deprivation by James Feng in 2019, also working with Mike Chi. And when they looked at the dynamical states in this, the functional connectivity, they found that there were like 5 states, including this task-ready state, as well as a low and a high arousal states. And they found that some of these, such as the task-ready state and the low arousal state, did change with sleep deprivation. And the amount of time spent in these on an individual level was related to deficits in cognitive performance. Now that's nice, that shows you like the repertoire of different cognitive domains, but if we stay on the track of integration and segregation here, what we can do is use a framework by Max Schein in his 2016 Neuron paper, where you look at graph theory metrics of within and between network measures, and if you track the the segregation over time, you can sort of cluster the brain into two extreme states or modes, the segregated and a more integrated one. And if you look at that over time, you can see all of the cortical regions fluctuating in what he termed the Cartesian profile of integration segregation. So on the y-axis you have the within network, and on the x-axis you have the between network. And you can see that the brain does fluctuate from integrated to segregated states. He showed that this was related to cognitive performance, and it has been theorized that for effective cognitive functioning, our brains need to fluctuate between moments of segregation and moments of integration. Now, if we look at the amount of time spent the brain spends in different areas of this 2D Cartesian plane, between the sleep-deprived and well-rested states, we see that there's quite a striking difference here. Now, before I explain that, I just want to say that it is related to deficits in cognitive performance, so there's a functional relevance to this. But basically, this is a meaty graph, so I'll try to explain it. Blue means the sleep-deprived state, there was less of this occurring, and yellow means there was more in the sleep-deprived state. And so you see that in sleep deprivation, the brain spends less time in the really highly integrated modes, and also less time in the highly segregated modes, and more time in this middle part. And that can be shown nicely when you split them into the integrated and segregated states and compare them. You see that the segregated state, that's less segregated in the sleep deprivation, and in the integrated mode, it's less integrated. Another way to view this is to look at, within the well-rested state, the difference between integration and segregation. It's quite striking. In the sleep deprivation state, there's less of a difference there. And this has an influence on the transitions. So there's actually more transitions in sleep deprivation and less time spent in each mode. And if you can think about this, basically when we're well rested and we're optimally performing, our brain is nicely fluctuating from one extreme of segregation to an extreme of integration. But when we're sleep deprived, the lines become blurred and our brain has trouble going to the extremes of each mode, and therefore there's instability and more frequent changes between these two modes. Now, the thalamus is actually involved in this, and when we switch from a segregated to an integrated mode, our thalamus becomes active, and it's less active when we switch from integrated to segregated mode. And what we found was the thalamocortical functional connectivity decreased in the sleep-deprived state. And so what effect this might have? Well, the thalamus is firstly part of the ascending reticular activating system, but it's also involved in facilitating the generation of intrinsic functional modes. So a decoupling of the thalamus from the cortex might have some strong impacts on this ability to switch from integration to segregation. Why is this important? Well, we know that when we fall asleep, the thalamus decouples from the cortex minutes before we actually fall asleep into an unconscious state. So again, this might be a homeostatic pressure mechanism that's decoupling and destabilizing the cortical integration segregation. Now, with my last minute, I'm just going to quickly show a clinical implication of this work through a model of narcolepsy. So narcolepsy is a disorder of vigilance, of hypersomnolence, and it is a neurological condition in which the hypothalamus, particularly the hypocretin cells, are affected. And the symptoms of narcolepsy are sleep-wake instability, excessive daytime sleepiness, and impaired cognition, so brain fog, impaired attention. We performed this study also in Montreal we had narcolepsy and healthy controls, and we gave them a bunch of cognitive tests inside the scanner whilst measuring functional MRI. And importantly here, we also did some healthy controls, and the healthy controls came back. They weren't fully sleep deprived, but they slept for 4 hours, so they were heavily sleep restricted. And when we look at the ratio of within to between segregation, we see, like in the previous study, when we're sleep restricted and sleepy, there is somewhat of an increase in this functional clustering, mostly in the somatomotor and the visual areas. But interestingly, in the narcolepsy condition, there's actually a decrease in this functional clustering, and it's localized strongly within the default mode network. Now, this is an effect of both the within and the between measures, but it's more strongly driven by the within. And what's interesting there is that it has been also shown previously that when we fall asleep, the default mode becomes decoupled within itself. So there's less within connectivity within the default mode network. So this may be an indication of sleep-wake instability in narcolepsy. What might be going on here as well is that when we are sleep restricted, again, we show that there's a decrease in thalamocortical coupling, but in the narcolepsy compared to the well-rested controls, that effect is there, but it's much reduced. Why might that be the case? Well, again, it could be a homeostatic pressure mechanism in healthy people that's not there in narcolepsy. What we do see is that there's an increased influence of the hypothalamus, or at least an increased correlation between the hypothalamus time series and the thalamus, so that we know there's a neurodegenerative effect or a degeneration effect of the hypothalamus in narcolepsy, and this is more tightly coupled to the thalamus in this condition. So to sum up, functional clustering of information is impaired in sleep deprivation and is related to cognitive deficits. This can impact the dynamical switching between integrated and segregated modes, which is important for cognitive performance. The thalamus is important in balancing integrated and segregated modes in the brain, but becomes disconnected under increased homeostatic pressure. And this is contrasted to neurological pathology such as in narcolepsy, where thalamocortical functional connectivity is less impaired and instead influenced more related to hypothalamus degeneration. With that, I'd like to thank all my collaborators in Canada.